All right. Can assume people hear me okay? Is the mic too far?
No, it's good.
All right, that's perfect.
Good.
Thanks a lot. Okay, so welcome everyone to today's the week 2 recitation. And if you attend the class yesterday, you see that we work a lot on the DNA write, edit, and read. So today we're gonna focus on the DNA work, and it's gonna be related to the lab that we're doing tomorrow for the MIT people here. For those who are doing the accessible lab or trying to set up things that I know this will be relevant as well, you can get into like the gel running workshop. So just to give everyone some heads up, today information is going to be, we try to make it more like a summary of what George and Joe have been talking. So there'll be a lot of information that we're throwing at you. We're going to get more technical in the way of like, how do we deal with this DNA? So we'll use the programs, so we'll see how do we order DNA through Twist Bioscience, where Emily was also showing you what the technology was capable of. And if there is any, confusion, we'll spend a few minutes during the class at the end of the bench lane just to make sure that everyone is on the same page. And also like at the end of Twitch, we'll just come back again. So please, if you have any question, feel free to drop into the chat and also like use those time to see what kind of confusion that we can resolve during our class. All right, just for the interest of time, let's move forward. All right, cool. So today is a recitation part. The part 1 is about the gel. And before we get into the gel, we're gonna take a look at what we are working on in terms of homework. So if you take a look at homework, there are 4 questions. We try to make them relatively general. So if you have no clue what's going on, feel free to use Google, feel free to use anything to like figure it out, what are we looking at. And the last question that we have is the most complicated one in our opinion, but it would be a good mental exercise just to get into like, if you want to think about any gene or any DNA or any protein, how do we start? So this question will, bring you to like, what kind of DNA you want to sequence and why? The right is also the same thing. What do you want to synthesize? There's some guidance around here, so please take your time. Again, it doesn't have to be extremely complicated. If you wonder what people have done in the past year, you can take a look as well. All the TAs that were distant from last year, they'll have this kind of homework in their system. But please, don't just copy it right away, because everyone should have their own interest, right? And the edit is like, something that was further along the way, because like when you can read and you can write, if you want to do just a small edit, just to make sure that, let's say if you want to edit like a bacteria or DNA, you want them to be the same bacteria. You can't just rewrite everything, right? So in that case, the small edit is something pretty important for us to work on as well. And not maybe like George mentioned something about CRISPR, and that we will look further into in the later classes. So that would be something interesting to look forward as well. And these are some papers. If you have no clue what we're working on, like try to look at these papers. The review is one of the scientific article that will provide you some initial information. If it's, not your type of reading, feel free to go into some blogs or like some web page. Like one place that would be great to go to is AdGene's blog. Like they're doing very great job at like introducing something simple to everyone. Just to get started, so we are working on the gel art. What is the gel anyway? So gel that we are referring to for the DNA stands for gel electrophoresis. So electro means electric, right? So we're gonna like move them using electricity. And when we do these like cut, copy, and join, these are something we're gonna use the gel electrophoresis as our tools to move things over. That's part of that. Before we get into the gel, if we have some starting DNA and we want to do some modification to them, one simple task that we can do, and it's technically like the first technology that we have in the genetic engineering world, is actually the restriction enzymes. The enzyme is something that can work biochemically, right? The restriction means like something that restricts their target to something specific. And in this case, is a restriction endonuclease, where it's going to find the DNA that have this sequence, GAA TTC. If they find this DNA sequence, it's going to cut it. So apart from just cutting, they also like have some pattern of cutting. So if you look at this, hopefully you can see my mouse for the Zoom people. When you cut them, there are two possibilities that you're going to generate this kind of what we call the DNA ends. This one we call them sticky ends because they have like some single stranded DNA that was hanging there that they can attach to other things, and the other one is blunt ends. So technically, the way that we cut these DNAs depends on the enzymes. Like this enzyme CoR1 will cut right here and make this sticky end. This enzyme SMA1 will cut right here. If you wonder what E. coli came from, it's actually E. coli. It's like E. coli, like enzyme 1, something like that. So in the past, that's how we get things to work. By having these enzymes, you can do what we call DNA molecular cloning. So if we have DNA and we want to move them like from place to place, you can technically do these molecular cloning techniques to edit or to amplify DNA from other places and put into the new place. One of the breakthrough technology for these genetic engineering, we call them recombinant DNA. And by recombinant, it means like we take some DNA from one organism, put into other organism. And that's considered recombinant DNA that can make recombinant RNA, recombinant protein later on. Even though these, if you like look into the textbook, these are kind of like one of the like primordial or primitive tools. It was used for making insulin. Like the first insulin, the first recombinant insulin that we use in our world, use this kind of technology to start with. And a lot of the techniques that you guys will be using later on, if you are into the biomedical research, you'll be using restriction enzymes. It's like pretty hard to like beat these kind of basic technology. So this is the chemical structure of DNA. Most of the time, most of the time your DNA will be a double helix. So whatever picture you've seen so far, it seems kind of fancy in double helix. And they have these base pairing where A and T will bind together and G and C will be bound together. The pattern is pretty consistent. If you have like A on one strand, there will be T on the other strand. This is how like biology sort of like replicate themselves. Use like A as a template, put T on the other end. If you see T on the template, you put A on the other end, and so on and so forth. So what we can do is that like if we have the DNA, if we cut them or we isolate them into different parts, we can isolate them by the size. So this is the simple picture from a team. If you do electrophoresis, you can push the DNA to move across these gel casting, like gel running system. When you put electricity from this direction, the DNA, which is negative charge, they can move from this to this end. So think about you have the same kind of power and you're just driving a car. If the car is so heavy, you're gonna go slow. If the car is pretty small, like Ferrari, it's gonna go fast. So the DNA that move, most of the time, the shorter ones or the smaller ones will go faster. So again, these are the negative charge that were highlighted right here on the phosphate group. And this is the reason why we can use this electrophoresis for kind of separation of the DNA. Keep in mind that you have the DNA that were very close in size. It could be challenging to separate this thing, right? Because they're the same size of car. No one knows if that car is a Ferrari or a Lamborghini or whatever. If they're very fast. These are like getting further into the charge or the mass or the length of this thing. These are 3 key parameters. Most of the time, we take a look at the mass because all the DNA will have relatively the same charge per mass. So if the mass goes up, then the charge per mass is like getting conserved. And that could refer to the length of the DNA. Most of the time, when you see these gels, they will have like these numbers associated with them. The number on the left-hand side on this DNA ladder here refer to the length of the DNA. So if they say, let's say 3.0, that's like 3 kilobase, so just 3,000 bases per. On the other side is probably the density of these. So you have a lot of DNA, the band is going to be brighter. If you want to draw something using this DNA pattern, you may want to consider if you want them to be brighter, you may want to duplicate that kind of cut size because like if the DNA gets smaller, then the weight or the mass of DNA they have kind of gets smaller as well. So that's why when you do the DNA cutting, you take the same DNA and you cut them into 2 pieces. the one that's bigger usually is brighter because they have more mass, right? All right. This is, hope you can hear the
freezes is the standard.
So this is like the video that you can show, that you can see like how the gel running actually work, and you'll be doing them in the lab at some point. So this is like the electrode, we connect them, and this is how they flow. This is not DNA, they're dye, so that's gonna tell us like how these small molecules are flowing, and DNA is usually bigger than those dyes. So if those dye doesn't fall off, all the DNA usually stay on the gel.
Which is a collection of DNA fragments of
So you just like go pretty quick on here, and you can take a look at this further. There are so some buffer or some system that you can like work through. Like if you use different buffer, different system, they can move differently, kind of. So again, that was a kind of funny video, because like there's some movement. They're using electrophoresis, and you may notice that the gel was moving as well, and that's partly because the gel that was like a little bit floating into the solution. If there's a lot of bubbling going on, then they can sort of like flow into the direction that they're going.
Or crispness of your DNA bands. You can use a gel comb.
Yeah, and this is something that is like one of the groundbreaking technology in gel running. Think about like you want to put something into the well, if you cast them without having this comb, it's so hard to pull them into the gel, right? And your band will not be kind of like straight or like easily distinguishable. So make sure if you cast a gel, you forgot the combs, don't try to poke them, just melt them again and put it back, or just add them. So yeah, this is very cool. And the final thing I want to say for the gel is that like the DNA doesn't have color. So we usually use some kind of molecules to attach into this DNA and make them like shine under the light. So most of the time we use the fluorescent dye so that they bind to the DNA and they'll just shine up once you give them the blue light. We use blue light these days, like in the past, like 20, 30 years ago, we don't have that technology yet. So we may be using with something that can sort of like get into your own DNA. We call them manipulating agent. Try not to use them. It's kind of a little bit scary. All right. Okay.
Any questions so far about this? I want to just pause real briefly there. This is also something that we're going to do in our lab tomorrow. So we'll have lab tomorrow from 1 to 4 p.m. We'll be running a few of these gels tomorrow at some of the stations downstairs in the other buildings. That way you guys can use this. And then this will be like a staple technology and staple method that you use for a lot of your experiments in future labs. You've seen some of the other protocol. DNA gel electrophoresis is probably like one tab of like, okay, now conduct a gel. And so this is one of the very key processes. So any questions so far?
Yeah, go ahead.
I guess I've always been a little bit confused on like connecting the gel and like why we're doing it, like with the friction enzymes. It's never been super clear to me.
All right, I think like restriction enzyme is something, for example, when you see, we ask the ChatGPT to tell us something about like, what is this? What is restriction enzymes? So there's some kind of secret that you can like cut specific DNA part and give you the DNA part that you want. So in the past, this restriction enzyme can be used to diagnose some disease. Like if you have some mutation in your DNA and they should get cut, then that can give you a different pattern from what you've got. And like something like, your DNA, like if you see like some forensic test in the DNA test, those are something that was a product of the restriction digest of your DNA as well. So we'll show you why you will need to use these restriction enzyme across the recitation, and maybe like if that's still not convincing to you, we can talk further.
I think another like interesting aspect is like we use them for a lot of stuff, but then this it's like we're just trying to show you why there's a use case for it. So like for example, for restriction enzymes, this is a very cheap and cost-effective way of determining whether you have like a correct cut. Like for example, a cut in the general area that you expect. Like let's say like I still show you how to do it in Benchling and online software, so you can predict exactly where this should cut based on what it recognizes as your restriction area. And then you want to go quickly check whether if it's like the correct area or not, the correct sizes. So let's say you cut like a 3000 base pair in half, right? You get one fragment that's, let's say we cut it in like 2/3, you get one that's 2000, one that's 1000. You should still see two bands that are now at 2000 and at 1000. So it's a very easy way to determine whether if it cut at the right place. And we combine both of them into this first lab. So that way it's an effective way to just show that way you understand what's going on, and then also use the technology in the first place. So
yeah, like to summarize those like response, they're very reliable. It's been around for almost 50 years. So that's why we're still using them. And a lot of them were just like widely available to the point that like you can just use them really easily. So just to bring back to our conversation or like in the biology for in general, let's say, this is like a schematic of like how the information usually flow in biology. We talk about DNA a lot, right? And technically, like the reason we talk a lot about DNA is because DNA is the way that biology was storing the information. Most of the time, the information that were stored in the DNA will just replicate itself, turn itself into an RNA, or getting turned over into the protein at the end, where this protein will just do some other cellular functions. It's not just protein, like sometimes the RNA do the job as well, but like it doesn't change the fact that like most of the information were stored in the term in the form of DNA. So think about like, if you have a hard disk, you want to give them to your friends. You usually just don't give away the hard disk, right? You just like convert them into something that is like easily shareable, put into the cloud, or just put into the thumb drive. I don't think anyone is using it anymore. Or at the end, it would just turn into the function. So what we care a lot in the biotechnology is that like we want it to be easy. It's so hard to like try to put a chunk of gene into yourself and try to like get a lot of those DNA. So we'll be using these like very useful DNA type called the plasmid DNA, where they're small and they're like pretty portable and pretty easily to put into the bacteria. So that's why like we're going to be talking about plasmid bio a lot in this class. So let's get into benchling. I want to emphasize that like we did the benchling deep dive in the bio bootcamp class number 3. So I'll show some demo, but it may not be as long as a 1 hour long benchling. So if you wonder what is going on, you can take a look at the slide or take a look at the video recording right there. So the reason that we have to use benchling is because if you take the DNA, most of the time, you get it in this kind of form, and it's not that readable, right? Like if you ask you to, hey, can you remember these, and like just keep working with them, or just open
That
them
would be
in
a
a notepad.
great final question, actually. Final project question, just like go memorize this.
Oh
That's-
yeah,
That's not actually happening, I was gonna say.
yeah. So if you take this, they'll be in different form. This is the fast A or FASTA, whatever you call them, and it's the GenBank file. So they will have like more annotation. I think you can guess that this format on the right has more information, because they seem to be formatted in a way that you can read them with some programs. And that's the program we're talking about. We're going to use benchling to see how we can work around these. Another thing that's very important is that like you see that they don't have a lot of denomination about like the 2 strand of DNA. So the program will also help you copy the other strand. And it's not a simple copy, right? It's the reverse complementation. So if the A is on the top, T should be on the bottom. So this program helps us a lot into exploring what it is. Before we get into the program, I'm going to show you where you find the DNA first. So if you wonder, like, you think about a protein, I want to know, like, what is the enzymes in your mouth that was helping, digesting whatever you were eating. You can Google them, but most of the time, Google will not show you the DNA sequence, right? So you can go into these databases. For example, NCBI is one of the good ones. If you're looking for insulin in NCBI, you see these kind of results, and they'll give you something resembling whatever data you've got, like in the previous slide. You can also go to UniProt, like prot stand for protein. And this UniProt can also give you something similar. So like this is also the same, human insulin. And these information usually link to each other. So you don't have to use all of them. If you use like one like widely used database, they will be linked to the other ones. And this is what you get when you get into the insulin from human. They have like some download. You can download these DNA and just keep using forward. Sometimes they'll give you the protein sequence and you have to convert them to the DNA, and that's part of the homework. So just make sure you can do it. All right, getting back to this like digestion, I'm gonna go fast now, but this is how you work on benchling to get things started. If you import your DNA sequence, which I'm gonna show you in the demo, you can sort of like go to the restriction enzymes panel. By doing that, if you ask the benchling, like, hey, I have these 48 kilobase DNA, can you try cutting it using this enzyme called EcoR1? And it sort of show you that they're gonna cut 5 spots. What kind of pen you get? So if you run digest, you get this kind of virtual gel run. They may not be exactly as what you've got in the gel, because like you can see that these ladder right here, they're kind of like linearly or like almost linearly comparable to like what you see. So the gel might be skewed a bit, but they're usually like the same thing. If you put the dot pattern into this DNA gel, you usually get a dot or like an elongated dot. So that's like what you're getting for the real thing. If you use another enzymes, which is equal R5, where they cut more, cut in more spot, you just get like, you know, a more complicated pattern right here. And that's where we go, right? One of the questions in the homework is that like, can you try digest these with different enzymes? So give it a try. And you can also like make this kind of pattern if you have like, you know, some pre-designed pattern that you can work on. These are the links that you can go into. And before we get into the showing, I want to show you something like what is the reason why we don't want to do this benching, or why do we not do this restriction ligation? So for example, oops. Is there any question from the Zoom?
Yeah, can I ask a question? How do you know which enzyme cuts where?
Yeah, very good question. So these restriction enzymes, as the name said, they're restricted to cut like a certain part. So in benchling, they will tell you what is the DNA that we recognize, that we restrict to that cutting. So on the tools, which I'm going to show you in the next few minutes, they will have like the pattern. Let's say they could be 6 base or 8 base, where they're going to be like the recognition sequence for the enzyme to go and cut. So if you wonder where they can cut, benchling is one of the tools that can show you where the cutting spot would be. Very good question. So this is an example that came from like one of the example I showed during the bootcamp. What if I have the plasmid that can express the protein, whatever protein, and in this one is expressing GFP, green fluorescent protein, that would make the bacteria go green. If I want to change it to something else, for example, make it red, like, you know, like the car has to stop, like we're going to turn the light from green to red. We can change this gene from the green fluorescent protein to the red fluorescent protein. One way to do it is to find some cut sites around here and just cut them, get rid of the GFP and replace them with the MRFP. So if you have these two sequences that can assemble them together, you can actually turn these, let's say when you do it for real in the tube, you'll be running the gel. And let's say I'm going to cut this part from the digestion, I'm going to cut this part from the PCR and try to combine them together, maybe the ligation. And at the end, we transform them. It's going to be something like this, where the starting material is green and you turn them into red. So that's the actual action that was happening. It's not going to be like this colorful if you're working on something that doesn't give color, but you can also do the reading to make sure that your edit was correct. So before going to the protein part, let's do a quick benchling showcase here. All right, I think the timing is still okay.
Yep.
I'm gonna do the Google and ask for L protein. Or maybe let's do our, let's go for insulin sequence. You know, AI is trying to tell me that, yeah, they're A and B protein, whatever. Uniprot came up as the first one. Let's go to Uniprot. And in Uniprot, they have a lot of things here, right? If you want to see what the sequence is, you just scroll down. And you see the sequence around there. That's all information.
That is a lot.
All right, I'm getting restless. Sequence. All right, here's the sequence. You can download this sequence, which is a protein sequence right here. If you hit download, they'll give you this kind of like FASTA file. Yeah, that's one of the way. If you go for NCBI database, you see this kind of information. There might be more of them. These are something that was more particular about these information. This seems more like a database. Going to here, you see more assembly and more things. These are the genome assembly of these particular information. Go for insulin. Then you get more genes, and this is what it shows up as a gene ID. Just take us back there. That's a file. It's gonna give you something like that. If you want to download to your computer, you can download here and just tell them what kind of format are you looking for. There are some GenBank that might be more informative. One cool thing is that if you take these reference sequence and go to this project, you can do import and tell them that this is the number I've got from database and try to search for it. And if the number or the denominator is correct, it's gonna try to access. Maybe this one is like an assembly number and may not be able to give you the actual information. Let me show you something that we are actually looking for, which is the lambda DNA that we're gonna do the digestion with. So if we go to this lambda, I think the description should tell us the accession number right here. So this accession number must be correct no matter what. If we try it again and import from this accession number, then it's showing up. So don't get too upset if the number is like weird and can't get you to the right spot. There are a lot of number that help you get access to this thing. Try a few, and if they give you the right one and they have this kind of like logo of where it come from, it should be correct. So this is like way more trustworthy than me copying, whatever sequence or type them down into the DNA. So this is what it looks in the sequence. You can see now that they have like the bottom strand as well because they're DNA, right? They have their double stranded. You can hit split workspace here, and it's gonna show you more information on this one. If there are a plasmid, if they are like something that was circular, they're gonna show you something that was like circular as well. So let's go to one of these examples. If I take my plasmid, they tell you that it's like some kind of like GFP containing plasmid. They can turn this DNA into the circular form, and you can sort of rotate them around. You can click at the annotation. This is one very important thing about these DNA reader. If you don't annotate them, it's almost impossible for the human brain to read through. Like, oh, I have to look for the ATGC, AGC, ATT, CAG, and that's gonna be some GFP. That's too difficult. So this annotation is going to be something that tells you whether like what protein or what information that would be. If I double click it, it's going to show like where it starts in this plasmid. What is the name? Is it a coding strand? Are they reading from the left to the right or the right to the left? That's forward and reverse. That's pretty important because DNA has two strands, right? So you have to make sure that you're reading at the right spot. Like for example, this is pretty easy to guess because it starts with ATG. If you have learned biology before, ATG is a start codon. So that's probably the right direction. All right. Let's try some kind of like ligation tool, maybe. So I told you that like we are trying to change the GFP into the RFP, right? So let's do assembly of the ligation. So if we start with the backbone that has this thing. It's not the right one, maybe. Let's try the GFP. Where's the GFP? This one. This one has GFP. And you can see that like there are some restriction enzyme that show right here. See that? Equal R1, KPN1. These are something that show you what the restriction enzyme is. If you try to search into it and see like if it's the only thing in the sequence, let's search for KPN1. This is where the cut site is. This is the 6 base that KPN1 is going to recognize. If I hit KPN1, we try to cut KPN1. And we click at another one, let's say BAM H1. It's going to show me where the other side is right here. So if we do this by trying to cut them, like click at BAM and also click at the KPN1 and tell them select this one, then you can get this sequence. And For the insert, let's get the MRP and see. I believe they might be similar. And they might be able to give you something similar. It seems like it doesn't have BAM right here. So you may have to go back and like choose the right one. But the idea is that like if you change them to the right one. I'm sorry, I thought it was right, but it seems like it shows the wrong enzyme.
I think also, like, even while finding these issues, you can see how easy it is to spot issues here. Like, if you're doing this manually, if you're writing this out as people used to do a long time ago, very error prone. And when stuff like this, like, for example, for me, for cloning, it can take up an entire day just to set up a lot of the reagents, like the PCRs, the assembly, etc. So this really saves a lot of time by doing the preparation ahead of time to make sure that whatever errors occur, either because, let's say, something happened with reagents, or it's just a major issue with your plasmid. So
Absolutely, yeah. So that's like, we call this process like in silico cloning. So you see that like they can stick to each other, but maybe not perfect. If you do reverse orientation, they seem to be better. And you can sort of like click yes, and they're gonna like, did you generate the new DNA right here? And this is kind of like how the new cloning can be done in the computer, something like that. Yeah, you see that this is an error that came, it doesn't have a backbone. So maybe like they have some other cut sites in there that leave the other side out. So this pair of enzyme is not workable. So before we go forward, I'm gonna show you one thing, because we use a few enzymes here. One sec, let me do this 1 minute showcase. So if we have these 4 enzymes and we try to cut them on the lambda, and we run the digest similar to what we show in our slide, the virtual digest will be like this. So it's going to show you a lot of things, but this is by cutting with 4 enzymes. If you do just equal 1, you get another virtual digest that have something more simpler by cutting only with equal 1. So this is an example of like how do you use the digest. It might be quick, but like take some time and play around with it. All right.
Sorry, I don't mean to keep interjecting. Document this. Like when you put it on your website for Notion, these kinds of stuff are critical for figuring out not only if you've done it right, but again, also debugging and just making sure that we know that you've walked through this process. So make sure you do this. And even for like committed listeners as well, definitely go through benching and document this portion.
Yep. All right. So want to get back to your question.
Yeah, I was wondering if you can go the opposite way of like, can you identify a sequence of like A, T, C, blah, 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 and then find the enzyme that corresponds to that?
Yes, but it might be a lot harder. So sorry. Still okay. Let me go to the sequence map and show you something real quick. So this is the list of enzymes that I have in my let's say my cabinet. If you go for all enzymes available, it's gonna show you a lot more stuff. You can also have an option to show only something that cut once or cut everything. If we show all enzymes, there'll be a lot of things shows up. Okay, my computer is processing because I have too many enzymes.
You can also see how many there are. Like the number of
Yeah.
cuts there, it's like 71 is a lot.
Yeah, so this is what happened when you try to like show all the enzyme cut sites. And as you can see, there are a lot of cut sites. But it doesn't mean that if I choose GTT, TTTC, whatever, maybe there's no enzyme for that kind of DNA sequence. So of course, you can look for some pattern, but it doesn't mean that you have the enzyme that was, commercially available for you for those enzymes. And that's why CRISPR or other things is groundbreaking, because you can program that. We'll get back to that later in the class. All right, just for the interest of time, I'll come back to this and move forward.
Any questions on the bench thing? Again, make sure you go through this at home. Like, I think for people that are doing the lab tomorrow, if you guys have time today, definitely go check it out for at least like 5-10 minutes to like get yourself familiarized. It always helps to go through and read the websites and so forth for how to even find the file. Oftentimes students get stuck there, but any questions on the bench thing so far?
Yeah, let's take a few minutes here and see. If you're still confused, which is completely fine, we can like come back and talk about this in the next recitation. I'll talk with your TA tomorrow as well. That's also cool. And maybe at the end we can like form a group and we can ask each other what's going on, just to check your understanding.
So basically, the idea is I have bench string and I then search for the DNA that I'm kind of interested in.
Yep.
I then load it in
Mhm.
and then I look for a specific point I want to cut, or like how does. And then once I know that, I look for an agent, or how does that?
Yeah, so it depends. So if you're looking for a specific gene that you want to put into another DNA and do molecular cloning, like what I showed, that I'm looking for the MRP, try to put into the plasmid that used to have GFP, green fluorescent one, trying to make it red. That's something you may want to do in the lab, and you will actually be
You will
doing
be,
in the
yeah.
lab, in the Gibson lab. So if you're interested in protein A, like part of your homework, and you want to put into something that you know, you can either go look for like if it's a human gene, maybe. I'm not sure about the ethics here. Please check it out. You can amplify the DNA from yourself and put it into the plasmid.
We
Theoretically,
used
that's
to do
possible.
a lab stuff like by doing like by testing for blood type. So like back a couple years ago,
That's
we used to
actually
like,
doable.
where you can get your saliva from your tongue and then just actually go ahead and like sequence it and do PCR in certain sections, to see which blood type you have.
Yeah, so those are something that people have done in the past. You know, you're interested in these, you know, let's say worms. You want to know what kind of DNA they have. You amplify the DNA, put into the plasmid, do the sequencing, and that's how you know what kind of DNA do they carry. If you know from the database that what kind of DNA you have, these days the DNA write, which can make the DNA for you without having to secure the original DNA source, they allow you to make those kind of like 1K or like something bigger to go in that direction. So this is one of the potential of the DNA write, apart from just like being able to read or being able to like, you know, do them in silico. You can do it for real. You can get the tube that has the DNA of your interest.
And for the purpose of tomorrow's lab, again, just like for in-person students, it's going to be focused on using actually a combination of different restriction enzymes to go just demonstrate that you're able to cut this DNA, get the size that you want, basically. It's not going to have any, like we call them downstream applications. So for example, items like PCR, assembly and so forth, that you can then use the products of this restriction digest. We're not going to be doing any of that for the sake of this lab, just to get you guys in the lab. So yeah,
All right.
I think that's
Let's move for this part for briefly. If you have any question, you have the slide and you can reach out to us for more question. We want to go for the central dogma a little bit, like to dive deeper, like what is going on. So as I show you that it starts from DNA to RNA and to proteins, the reason that we care a lot about like the information flow is that we want to make sure that you get the right protein if you design at the DNA level. It's not trivial. You have to make sure that they're going right. So when they do this, they just transcribe from DNA to RNA and make the protein. The process that make proteins, they have to do some kind of like sophisticated conversion from the DNA into protein, right? Because they're completely different molecule. For the DNA and RNA, they just have like a few different bases. They may have like, you have to turn from A into U for RNA, but that's still kind of like simple rule. For the DNA to protein, that's kind of difficult. So in human and bacteria, they have like something that was pretty conserved, in which they turn the DNA, what kind of DNA it is, into the actual protein pretty easily. And the reason that we care more about DNA, about bacteria when we make the plasmid is that like it's so easy to make things in bacteria. Think about like how long you have to grow like some organism, let's say a plant. If you have to grow a plant from seed to like the actual plant, it takes like weeks or maybe like years even, right? So bacteria can do that overnight. And then copy a lot of DNA that you try to put into them and turn that into a million copy of those. So for the plasmid, there'll be some part that like is required to make sure that you get the right thing. Like they have to make sure they have something that they can replicate themselves on. They have to get some selectable marker, for example, like antibiotic. If they grow into antibiotic we put in, they probably have like antibiotic resistant gene that we put onto as a plasmid. And we may have other restriction sites that we can cut that out and put our new genes in. So that's why they are so informative. And this is a simple way of like how the bacteria replicate the DNA. It's just gonna like start at the origin and double them every time they grow. So at the end of the day, you start with one cell bacteria and turn into it, like put them into the tube. And at the end of the day, you may have like billions of them. You actually clone the DNA from one copy to billions of copy. And that's why we kind of call them molecular cloning, because we clone the DNA part.
Does anyone know how fast DNA replicates, like, or like doubles? Every how many minutes, hours, so forth? Does anyone know?
Can you guess?
Like 20 minutes.
Yeah.
20 minutes, which is like the same time that the bacteria needs to divide. So every time they divide, they double their DNA content, because all of them need the DNA to carry the information to express into RNA and protein, so on and so forth. So another thing that would be interesting is the PCR, and maybe like we'll cover that. Are we gonna cover that somehow?
What, PCR?
Yeah,
We're not covering it tomorrow, no.
okay, but we come back to the PCR process later, because it's kind of like very useful process. But like, if you're interested, you can read into these like information that we
It
provided.
does not hurt to look at it early,
Yeah.
like seriously.
Yeah, the most important thing we want to show you here is the codon system. So in the DNA or RNA into protein, you will be translate the information using this codon system. So the 3 nucleic acid will be corresponding to 1 protein. And if there are AUG, there'll be the start codon, which is most of the time the starting of the protein. And to make this polypeptide, you just have to read in the frame to make sure that you get the protein of interest. If you make the RNA and they miss, like, let's say one of these guanidine, this G later, it's gonna be a disaster. You know why? Because not just this information will be bad, the downstream position will be bad as well. So for example, these are the 4 base, right? And this is a terminus, and UAA is one of the stop codon. If you get rid of this G, this is not gonna be UAA anymore, because they're gonna take U to here and read them as a new amino acid. And the next one is gonna be AAG, and that's not a stop codon. So the protein will turn from 3 amino acid to something, who knows, like whatever long. So that's what happened when you have like mutation, when you cannot make the protein of interest, they turn to something else because of these, you know, kind of mutation. So if you delete or like add 1 base into the system, it's very deleterious. If you change 1 base into the other, you're affecting that amino acid, but you're not affecting the rest of the
I
protein.
remember we were so worried about this. So last year we were designing a protein for the course. I remember we were so worried about that, just like having a mutation or a frame shift, which is what called, that we had stop codons like interlaced between. We had like some extra,
Yeah,
like
yeah.
at least dozens of stop codons that
Yeah.
are placed so it's like not only next to each other, but also spaced like 1 or 2 amino acids just to try to prevent this.
Yeah.
Because we were that concerned. So
Yeah, and this is how it looks. So, you know, combinatorially, you have 4 nucleic acid, and you have 3 position. It's like 4 times 4 times 4, that's 64 possibility, right? So there are multiple codons that represent the same amino acids, and it's like based on like some kind of like complicated system right there. But keep in mind, you know, like you don't always have to use the same for 1 amino acid, and we'll see why at the end. This is how the tRNA sort of like carry the amino acid into the ribosome, which is our factory of making protein. They're gonna find their matching partner of the messenger RNA, like, and then try to thread these beads of protein together. And at the end, you just see, you know, what kind of RNA do we want to use. So as you can see that like there are different types of codon that you can use for each amino acid. The reason that we may want to prefer one over the other is the abundance of the carrier of those tRNA or whatever. So if they have a lot of like these CGC over the CGG that translate into this ARG amino acid, it might be better to keep using this code rather than this one, because that means that the availability of these tRNA, the carrier, is more abundant inside the cell. So if you keep using the low abundant codon, they'll have to wait in line before they can get those tRNA to carry the things. Question?
So if you do that, it's just slower, right? Like it doesn't.
Yes,
Oh.
yes. But if you want to make a lot of protein, like there's a lot of like biochemists who want to make a lot of protein. Let's say you want to make insulin. Instead of having like 1 gram per round, if you can make 50 grams per round, that's a lot better. And sometimes if it's too bad, it is too slow, they may not make it. So even though we see them as like being able to make Most of the time, codon optimization is something that sometimes not just change, 0.1 to 1. They can change like 0.1 to millions if they're much more efficient. So there'll be some programs that help us with the codon optimization. So the workflow, again, just to get back to how we work on, if we were to make a new DNA, we can design the DNA that we want, like from the protein or from the DNA, put them into bacteria, make a lot of copies of these DNA, isolate the DNA out, and try to put them into the new cells, whatever again. If we want to work on a cell-free system, we can just design a plasmid, make the DNA, and just put into the system. This is like added because like we have some kind of like reason that we want to use cell-free, and it's gonna be explained more in the cell-free system. So we sort of like can skip some steps if we use a cell-free system. So this is something that you can look at. This is probably the last slide before we get into the
Could we
technical
go
order.
back 2 slides?
Yeah.
So, one more slide. This workflow. So this slide, for all of you guys, this is a very critical slide. And I say this because understanding this workflow is essential for basically all of our labs. And this is exactly how we process through, like, for example, the Gibson assembly lab. So if there's anything you can get out of this, this is the workflow you want to know and use for the rest of the course, and even for your final project too. We work through a lot of the similar techniques to be able to get a gene of interest. And I was explaining actually a few of those yesterday. Ideally, a lot of these stem from the challenges we have a gene of interest either from nature or from other items that people have identified, and we want to put it into a chassis or an organism of interest. So this is the workflow you want to know.
Yeah. So to be, to explain more is that like put DNA into cells. This then doesn't have to be E. coli anymore. But this is usually E. coli because it's so good at making a lot of DNA. It grows so fast. So we find it to be a lot easier to do the cloning inside E. coli. One step that might be missing here, which is usually part of this step, is the sequencing. You may want to read them first to make sure that it's correct. Like there are some functions that you can use to make sure that they're correct, but usually reading is the most powerful way of making sure that they are right. So again, like, if you do molecular cloning, you usually like do the ligation that I show you in silico, try to make some DNA, put into the plasmid, and this plasmid will get into the bacteria, and we grow them and make the protein of interest. This is kind of like the process. If you want to get the protein, one thing that we have to do in this class is that you have to order the gene that you can make the protein interest. So we're going to show you, briefly how this process works using the Twist system, which is our partner. So if you go into Twist, you have to make your ID first. So I'm going to show you briefly. You can make your own account and just work forward. If you go there, you see this like ordering system. Let's say I go for genes. I'm gonna start a new project. I call them L protein, which is the protein of interest in our phage project. They'll ask you like what kind of genes you want, and it actually like depends on what kind of application you're looking for. You can ask them to do clonal genes where they put into a plasmid and give you the plasmid. You can go for the fragments, and you just get the fragments that you can put into the plasmid by yourself, or they can give you a mixture of everything that has some mutation, for example. So we usually go for the central one, like the one in the center, so that we do put into the plasmid of interest. The reason is because like the genes, you have to do work with them for a while just to make sure that they can put into the right plasmid of your choice. So if you order them as a gene fragments, you can put into the plasmid of your choice right away because you have that in the lab already. They'll ask you like, how do you want to get started? You want to start from the nucleotide sequence or amino acid sequence? If you don't know the nucleotide, but you know where you want to put it in, you can put the amino acid sequence and do the reverse translate into the DNA. Or if you want to just like, just work on the DNA that you have, and because there are some reasons that you prefer the DNA from the native organism over the optimal one, you can just go for the manual import. This is another tool from Twist that can do codon optimization for you. So the way it works is that like they ask you what kind of sequence you're looking for and what is the DNA. So you give them the DNA, let's say I like push forward and show the DNA. They're gonna ask me what kind of organism I want to optimize them into. And right here, I choose E. coli because we want to express them in E. coli. And when I run them, this is like another one. If you get back to like the order, once we get the DNA, this is the L protein that was already optimized, let's say. I just put that optimized DNA sequence, put it back here, and just do the sequencing. Just do the synthesis. And then they'll just get shipped to you. And hopefully you get that at the end. Wait, it doesn't go anymore. Maybe that's because it's the last slide.
I think so.
All right, so before we adjourn, let's give me like 1 or 2 minutes to show you what it is exactly.
And again, we'll walk you through this process when you do it. So most likely you'll be ordering clonal genes, or you'll be ordering like oligos as well. So oligos are like single-stranded DNA that you use to as like primers for PCR. Again, terms you might not understand now, but single-stranded DNA, you'll order either double-stranded DNA. Both of those can be ordered through Twist and other vendors. So we'll walk you through that. It's probably in March, I think.
Wait, the costs are all covered by the course, right?
Yep, don't worry about that. Yeah, well, that's one of the benefits
Yep.
with the, especially here, is like
Mhm.
Twist is covering a lot of the costs for this, so please do take advantage of this.
Yeah.
If you have a gene that you really find in
Yeah.
nature that's very interesting,
Mhm.
this is your time to do it.
Yeah, so I'm just like going to click as I shown right there. I'm gonna show you the different process. Let's go for amino acid. If you want to start with amino acid, and we wanna get the sequence. In this case, if you give them the amino acid, you may wanna tell them what kind of optimization you wanna do. This is sort of like an in process or in system code optimization if you start with amino acid, because they don't know what kind of DNA you wanna get, right? So let's say coli. Put that in. And if we go to my bench link, oops. Go for L protein. Is there any L protein here? Maybe
I
it's
see,
in
there's
here.
looking the cell-free ones, yeah. So honestly, we might be fine.
Yeah, I have this L protein right here. Unnamed protein, L protein. I'm gonna give them the protein sequence. So this is might be a little bit quick. I'm gonna show you again. I do the right click. I try to copy. And they give you the choice whether you want a DNA that you are looking at, the reverse complement of DNA, or the translation. So it shows the translation. Go back to Twist. Put that in. Name them L protein E. coli. Then continue. Then they're doing something on their end. And this is telling you error,
It's probably too short.
too short. So sometimes if it's too short, you can't synthesize them. And if you give them, let's say you have the DNA of interest already, right here. And you kind of like, you want them to be longer, you can sort of like put other DNA on top of them, and then you can take that to do the PCR later on. So for example, if you want to do the trick, like, I want to make them synthesizable, I may just add another DNA here. I did Control-C, this worked just like Microsoft Word. And add some DNA right here. Seems like that's not very straightforward.
Could you maybe add?
I haven't used them for a while.
Worst case, you might just go back to the first menu.
Yeah. So in that case, I can just go back and like turn this DNA into,
Really surprised they don't let you just add bases.
yeah. Huh, interesting. That's why we need benchling. So you can copy this, you can download the sequence as a FASTA file, put them into some other format, or you can just like copy them into benchling like this. Like in benchling, you can just like let's assume that that's the thing I copy. And just come back here, make them longer. And just take the whole thing. I think 600 should be long enough. I think their limitation is around like maybe 300 or like 500.
300 is minimum.
Yeah. And then,
Worst case, you might have to go back to the start.
yeah, let's just go back to the start. Redo.
Will all these links be posted?
Yep, so as soon as this is done, we'll post the slides on the website,
Mhm.
and then the recitation link will be up sometime today or tomorrow. But
Okay.
yeah, you'll be able to go through. We have links hyperlinked to the Twist stuff, so yeah.
Alright.
And Benchling's also free too. Benchling's
Yeah.
I think there are other tools that exist on like, I think Snapchat and other stuff that people would like to use, but eventually we use it because it's mainly free and has a good amount of features for us.
Yeah, so sometimes you get this kind of error. So the error that comes up, sometimes they will say like there are some problematic area. Or there might be some repeating region detected, and you know why it's repeating. It's just randomly like put the same thing onto it twice. And the way of the synthesis of these things, you know, you can't have too many repeating sequences. So this is not the original problem anymore. And you just have to figure this out and like find some ways to reduce the complication or the complexity of DNA.
I'm curious,
Also,
could
you
you click
cannot
on the
automate.
problematic area real quick, like the second one?
The second one,
Yeah, one more, hello?
this one,
What is it saying?
repeats or
Oh,
extremely
okay, that's a
low,
good one.
high, low GC. So they will have some scoring. So if it looks like it's gonna be very hard for them to make it for you, they will say like, nah, you're not gonna have your DNA, you're gonna have something else. So it's better to not do it. And these are some in silico work that can help you move forward. Like if they say that it's not gonna work, then not use it, right? All right.
So that's basically them saying.
Yeah.
We probably can't do it, we'll have too high an error rate.
Yep.
Like, we're not gonna, like, we can't buy it.
There could be a wide variety of reasons too, like,
Can you
Yeah.
force them to do it? Anyway?
Is it again?
I don't think you want.
So there are two categories. So there will be the one that would have high complexity to the point that you cannot order them because they're very confident that this is gonna fail. Then you can't order them. There will be some that say high complexity in the region that you may have some mutations. They'll let you order. But be aware that you may have some deletion or some mutation in your sequence because it's a difficult synthesis. Again, keep in mind the DNA, right? In the lecture that you were looking at, there are different ways of making DNA. So if it shows here that you can't make it using the twist platform, there might be other systems that can make it because they use a different. I'm not saying there's any like, you know, some perfect synthesis platform, but at the end of the day, if you have to like break them one by one, you can do it no matter what. So some solid phase synthesis or something that like, doesn't involve a lot of like repeating region, you can do it. It may not be as fast. So that's sort of like, you know, maybe like take away that if you cannot order it, it doesn't mean that it cannot exist. There's a lot of human gene that was in our body that, you know, the platform can't make. But we've been making it every day, you know, when we have moods, they just like fix themselves, they just duplicate. Because biology is way more complicated than we thought.
Yeah.
All right, I think that's all for the recitation today. If you want to stick around to ask question or like you want to group up to ask question, like talk with each other.
We'll
feel free to-
be sitting on it.
Yeah, we'll
Also,
be sitting
can
on
people
it.
fill out this attendance form?
What's the code?
Yeah, it's good.
Oh, I should have. How to go is fine. I'd pick a random one, but I am not very creative at the moment, so feel free to put anything. Yeah.
I just put it in like slide.
Perfect, yeah.
Like
We do that
tomorrow,
all
let's-
before we get to.
All right,
How
so
long is it?
I'm gonna say goodbye to the people in the Zoom real quick because I'm gonna end the recording right now. But thanks a lot for attending online. Again, like what I show you clearly identify that like nothing in your design could be perfect, right? You see me struggle through this process, even though I work in molecular biology for 5 or 6 years already. So don't worry if you struggle. I still struggle, and I did show you online here that I'm struggling. So please take that as like another suggestion. All right, I'm gonna end the recording right now. The Zoom is still open.
And see everyone tomorrow morning, you guys, I know. Or tomorrow.